Introduction. Indels, short for insertions and deletions, or genetic mutations that involve the insertion or deletion of one or more nucleotides in a DNA sequence. These mutations can lead to changes in the genetic code, potentially affecting the function of the resulting protein or causing other genetic variations. Insertion deletions are a type of chromosomal abnormality in which a DNA sequence, either one or multiple nucleotides, is either inserted into or deleted from a genetic sequence. Insertion. Insertion means that a number of nucleotides have been erroneously added to the genome, most often during the process of DNA replication. This number can be as small as a single nucleotide or up to thousands or even millions of nucleotides. The effect of an insertion likewise varies. Some may cause no effect at all, whereas others, even single nucleotide insertions, can completely disrupt the function of a gene and lead to pathogenic variants associated with a genetic disease. An insertion, as related to genomics, is a type of mutation that involves the addition of one or more nucleotides in a segment of DNA. An insertion can involve the addition of any number of nucleotides from a single nucleotide to an entire piece of a chromosome. In this, a nitrogen base is inserted or added to the sequence. It causes the triplet frames to shift. It always affects the amino acid and consequently the protein. In the normal DNA, CGA codes for alanine, TGC codes for threonine. In the mutated DNA, after the alanine, due to the insertion of A means adenine, it changes to leucine. Okay. Deletion. A deletion, as related to genomics, is a type of mutation that involves the loss of one or more nucleotides from a segment of DNA. A deletion can involve the loss of any number of nucleotides from a single nucleotide to an entire piece of a chromosome. Deletion mutations are actually the cause for a large number of genetic diseases, such as 2/3 of cystic fibrosis cases and the cat cry syndrome, which is so called because children with this syndrome often have a cry that sounds similar to a cat meowing. In this case, a nitrogen base is deleted or removed from the sequence. It causes the triplet frames to shift. It always affects the amino acids and consequently the protein. In normal DNA, CGA codes for alanine, TGC codes for threonine. In the mutated DNA, after alanine CGA, due to the deletion of G, guanine, the TCA codes for serine. In conclusion, indels can have various consequences on protein synthesis, gene regulation, and overall genetic function. The impact depends on factors such as the size of the indel, its location within a gene, and whether it results in a frame shift mutation or alters protein coding regions. Thank you.
Thank you, Lalasri. Next, yaro, presenter.